Hello friends, today I am here with third and last part of respiration in plants of 11th standard. In today's session, we are going to discuss about tricarboxylic acid cycle, respiratory balancing, difference between fermentation and aerobic respiration, amphibolic pathway, respiratory quiescent. First of all, we will start the session with TCA cycle. Also it is called as citric acid cycle or Krebs cycle. This cycle has been named after the German biochemist in England, Sir Hans Krebs, who discovered it in 1937. It is also called as citric acid cycle due to one of the participating compound. It takes place in mitochondrial matrix. Krebs cycle or TCA cycle, that is tricarboxylic acid cycle, starts with condensation process. In this process, acetyl coenzyme A reacts in presence of water with oxaloacetic acid, which is the normally present in a cell, and it will form citric acid and releasing coenzyme A for reuse in the pyruvate oxidation. This reaction is catalyzed by the enzyme citrate synthetase. Then it is followed by two successive steps of decarboxylation, which leads to the formation of alpha ketoglutaric acid and succinic acid. After that, succinic acid is converted into malic acid, and malic acid is further converted into oxaloacetic acid to continue the cycle. During conversion of alpha ketoglutaric acid to succinic acid, one molecule of GTP is synthesized. In a coupled reaction, GTP is converted into ZDP with simultaneous synthesis of ATP from ADP. At 3 points in the citric acid cycle, NAD plus is reduced to NADH plus H plus. The 3 points are: first is during the conversion of citric acid to alpha ketoglutaric acid. Next, during conversion of alpha ketoglutaric acid to succinic acid. And at last, during the conversion of malic acid to oxaloacetic acid. Besides this, at one point of citric acid cycle, FAD plus is reduced to FADH2. This is occurring during the conversion of succinic acid to malic acid. In addition, it also requires regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The total summary of the reaction is given here. Pyruvic acid plus NAD plus FAD plus 2 molecule of water plus ADP plus inorganic phosphate combined all together, and they give rise to 3 molecules of carbon dioxide, 4 NADH, 4H plus, FADH2 plus ATP. And these reactions are taking place in matrix of mitochondria. This is the flow chart of citric acid. You may locate the points where NAD plus is reduced to NADH plus H plus, and you may also observe here where FAD plus is reduced to FADH2. Now let us discuss about respiratory balancing. It is possible to make calculation of the net gain of ATP for every glucose molecule oxidized. But in reality, this can remain only a theoretical exercise. This calculation can be made only on certain assumptions that there is a sequential orderly pathway functioning with one substrate forming the next and with glycolysis. TCA cycle and electron transport system pathway following one after another. The NADH synthesized in the glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. Only glucose is being respired and no other alternative substrate are entering in the pathway at any of the intermediary stages. But these are the assumptions and are not really valid in living system, and all the pathways work simultaneously. Now let us see the differences between the fermentation and aerobic respiration. In fermentation, there is partial breakdown of glucose, whereas in aerobic respiration, there is complete breakdown of glucose into carbon dioxide and water. In fermentation, the net gain of ATP is only 2 ATP, whereas in aerobic respiration, net gain of 36 ATP. In fermentation, NADPH is oxidized to NAD plus slowly, whereas in case of aerobic respiration, NADPH is oxidized to NAD plus vigorously. Now it is about amphibolic pathway. The breakdown of respiratory substrates provide carbon skeleton for the synthesis of a large number of essential plant products such as polysaccharides, proteins, fats, nucleic acids, pigments, cytochromes, etc. Thus, the same process which acts as catabolic pathway for respiratory substrates also act as anabolic pathway for synthesis of various intermediary metabolic products and secondary metabolites. So, thus the respiratory pathway acts both catabolic as well as anabolic pathway. So, it is better to call it as amphibolic pathway rather than to call it as catabolic pathway. Here in the given flow diagram, it is shown about interrelationship among metabolic pathways showing respiration mediated breakdown of different organic molecules into carbon dioxide and water. Now, the another topic is about respiratory quiescent, that is RQ. Respiratory quiescent is the ratio of the volume of carbon dioxide released to the volume of oxygen taken in during the respiration in a given period of time, and it is taking place at a standard temperature and pressure. Mathematically, it is expressed as RQ is equal to volume of CO2 evolved to volume of O2 consumed. The value of RQ depends upon the type of respiratory substance used during the respiration. When carbohydrate such as hexose sugar, sucrose or starch are respired in presence of oxygen, the volume of CO2 evolved is equal to the volume of O2 absorbed. Thus, here the value of RQ is equal to 1 or unity. Here it is justified with the help of an equation. Here you can see that the amount of CO2 evolved and O2 absorbed are same. So, RQ will be 1. But when fats are used as respiratory substrate, then RQ is less than 1. And when proteins are respiratory substrate, then the ratio would be about 0.9. Now, the last topic of today's session is about electron transport system. Electron transport system is the metabolic pathway through which the electrons passes from one carrier to another, and it is formed in the inner mitochondrial membrane and aids in liberating and using the energy stored in the NADH plus H plus and FADH2. Electron transport system is a stepwise process. In the first step, NADH plus H plus which is formed during the citric acid cycle and glycolysis is oxidized into NAD plus with the help of enzyme NADH dehydrogenase, or also called as complex 1. During this, electrons are produced which are conveyed to ubiquinone via flavin mononucleotide. Similarly, the complex 2 or reduced form of flavin adenine dinucleotide or FADH2, which is synthesized during citric acid cycle, is conveyed to ubiquinone. Then from ubiquinone, electrons are accepted by the complex 3 or cytochrome BC1, which further gets conveyed to cytochrome C that serves as a mobile carrier between the cytochrome C oxidase complex and complex 3, comprising of cytochrome A and A3 with copper center, that is complex 4. During this transfer of electrons from each complex, simultaneous there is production of ATP from ADP and inorganic phosphate with the help of enzyme ATP synthetase, that is complex 5. This amount of ATP production depends upon the molecule that has been oxidized. Altogether, 3 ATP molecules are generated by the oxidation of one molecule of NADH, while one FADH2 molecule upon oxidation produces 2 ATP molecules. Here it is the flow diagram of electron transport system. So this is all about today's session. Thank you. For any kind of doubts, please write your question in comment section.